Okay, thank you, Philippa and Lee and Aurochs, of course, for giving us the opportunity to be included in this nice schedule. So I'm from SVI, from the Huygens software. That is the software that we develop, and we exist for more than 25 years now. And we started with deconvolution, and we are heading towards, we are already busy also for years, with restoration and analysis. And I'm very happy with this previous talk about the point spread function, which saves me some time in explaining that. So let's go further. So I think every user, every microscopist asks the question, like, how reliable is my experiment? I have my sample, I put that sample under my microscope, and at the end, maybe with some use of software, I get some numbers out or some nice pictures. So how am I sure that this is giving me the real representation of the object? Well, it's important, also mentioned in the previous talk, that you are aware of the problems that happens during a microscope acquisition. So let's just present you this picture here. This is an image of a wide field microscope, and it shows you beads, the 170 nanometer tetraspec beads that also were mentioned in the previous talk. So they nicely show the blurring cones. But in the same picture, you also see noise, you see hot pixels, you see chromatic aberration. So the green channel is a bit below the other 3 channels. So this is a multi-colored tetraspec bead, so 4 channels in fact. So there are lots of problems in this image, and this is a perfect image, don't get me wrong. This is a perfectly acquired wide field data set. We got multiple data sets from CytarGo, from different microscope types even, and they all look the same. So don't worry, a perfect image. And if we can run that through Huygens and fix these problems, and we can get this result. So I hope you agree with me that this image looks much more like the 170 nanometer beads that we have under a microscope. So what is true deconvolution, Huygens true deconvolution involving? It's a reverse process of a convolution. So every object that you have under your microscope and that you image with your microscope is subjected to aberrations, and you cannot go around that. And it can be partially modeled into a point spread function. And this point spread function then is the building stone of your sub-resolution object. And so every point source of light is replaced by a point spread function, and this gives you ultimately the image. So the image is just a representation of the object. It's not the real object that you have under your microscope. Although with deconvolution, Huygens true deconvolution, you can reverse this process and use your image with a point spread function and get to a representation of the object, a better representation of the object than you had in your image. An important factor here is that the sum of the point spread function, and that is what is the case in Huygens, the sum of the point spread function in terms of intensities is 1. That means that we don't effectively add or remove any terms of any signal really from the image itself. So the energy is kind of conserved. And then I'm neglecting background subtractions and noise corrections, but basically you should not add or remove any signal from the image. So the point spread function can be made, can be calculated, let's say, from the microscopy parameters. We have many different models for almost all types of microscopes, and I will present that in the rest of this talk with some examples. So we can build a point spread function based on the model of your microscope, and that is a very accurate model in fact, and that is what we can use for deconvolution. We can also use our PSF distiller, and this PSF distiller can in fact extract a point spread function from a bead image. This bead image can have multiple beads, and you can also load multiple images with multiple beads or single beads, that does not matter. What we have is a very nice wizard that classifies the quality of the beads in the image. So it looks at neighboring beads, at beads that are at the edges of the image, beads that have not a good intensity, and so they are being discarded. That everything is done by the Huygens PSF distiller. So the advantage is that you can have a very low noise image and still extract a good PSF from that. So this all boils down to having good analysis of your image. And what's the advantage of using restoration, deconvolution for analysis? Well, here's an example of that. Kinetochore complex from a human cultured cell, stained by fluorescence. On the left you see the raw image, and on the right you see the deconvolved and chromatically corrected image. So let's focus on this area, and if we load that into our Huygens object analyzer, we see on the left a segmentation, a default segmentation based on an algorithm called OTSU here. And you see that the objects are somewhat misaligned, at least. I assume that these should overlap better, and that is in fact the real case. But you see that it's not greatly segmented. If you look on the right, deconvolved and chromatically corrected image, and you see that the image almost immediately is properly segmented. And you can test the validity of that with beads, for example. If you deconvolve similar type of beads that are kind of treated in the exact same way as the sample, you can kind of validate this result. So the big advantage is of this that you don't have a subjective segmentation. You don't need to manually threshold things. You don't need to assign pixels if they're true or false signal. And after deconvolution, you can have an image that you can objectively segment with an OTSU algorithm or with any other type of algorithm. And that already gives you a good segmentation of the object. So some other examples. So here's a wide field data set of a dividing cell. On the left, the raw data, nicely acquired. On the right, deconvolved. And if we focus in on the pericentric staining, which is a staining that surrounds the centrosomes, and we make a line profile through that and plot that, then we see that the raw data has the red curve, and the green data, the green line, is the deconvolved data. So please notice how much signal you can gain by deconvolution, which really helps you with the segmentation of the objects. And also you see a lot better that this pericentrin is really around the centrosome and not also within the centrosome. So that deconvolution helps you with the segmentation, I said. So it gives you more trustworthy analysis. So if we look at the dendritic spines, for example, this is a confocal data set, raw, deconvolved. We look at co-localization at the synaptic area, and you really can distinguish the co-localization regions. You're not able to do that on the raw data. Here is a nice confocal data set from Benjamin Glick. These cells, you're trying to track objects like, for example, Golgi markers here, and you nicely see this time-lapse movie. Here is the raw data. It's impossible to track these type of objects from the raw data, and this is Huygens deconvolved. So we were very happy with these very low signal results from Benjamin Glick that could be nicely restored by Huygens. Another example here, ARIScan, restoration of PML bodies done by Peter Hemmerich and Christian Heuschen. On the left here, confocal data of the 32 detectors of an ARIScan microscope. And here is the rendered image. Here the ARIScan deconvolved data by Huygens, and you nicely see the donut structure of the PML bodies. So with ARIScan, for example, we can go down to 90 nanometer resolution, which is unprecedented. And this is what we've detected with the use of Catacomb beads. STAT3D, some examples here, mitochondria with, in green, the location of newly synthesized proteins So this could only be determined by Rolando Berlinger Pelmini and Matthew Zargal. Here, another example from STAT. And I very recently, I think it was yesterday, I received some data from Philippa. Aurochs Clarity. This is a sheep muscle section. It's commercially available, stained for different objects. This is the raw data, and here is the data deconvolved by Huygens. So Huygens, we offer true deconvolution, energy preservation, and we really model the point spread function for the specific microscope type. We have additional restoration tools like stitching, chromatic aberration correction, PSF distiller, and this all gives you very reliable, trustworthy results for visualization and analysis, tools that we also have in our software. And with that, you may hope to reach a cover of any of these magazines like one of our customers, like some customers have. So at the end, quickly, how can you use Huygens? So we have essential, professional, and localizer as different packages. Essential and professional, focusing on deconvolution and visualization analysis. Localizer for single molecule localization microscopy. They can now all be used everywhere. You just simply log in with your user account that you can purchase at svi.nl. Log in and use Huygens everywhere. So you can have Huygens installed on multiple computers, even at work, at your home. You can work in a train if you have an internet connection and can work. And even multiple users can use Huygens at the same time.
Brilliant. Vincent, sorry, we're you're at the end anyway.
So please visit our website, ask for a test license, try it yourself on your own data and see what we can offer you.
Brilliant. Thank you so much for that. And I'm really pleased that you managed to access work with our data at such short notice. So I literally gave Vincent this data set with minutes to spare. So
after my request yesterday.
Thank you very, very much. We'll send you some more data to play with over the coming weeks. So if you wouldn't mind, Vincent, there's a question or two popping up in the chat window, but we are running out of time for this session. So do try out this software. It works with our data, and as you saw, it works with a wide range of different disciplines. So it's a very, very powerful and well-known algorithm.